Okay, so this is a video on digestion and absorption of lipids. So there are two revision tasks. The first is to describe what micelles are and how they're formed during digestion. And then task 2 is to describe the complete process of digestion and absorption of lipids. And I want you to refer specifically to organs, the enzymes, and the transport processes involved. So pause now and test yourself on those, and then we'll go through the answers. Okay, so the answer to the first question is here. So important keyword there, emulsification. So that's the formation of those small lipid droplets, those micelles, by emulsification by bile salts. And this is important in increasing the surface area on which the lipases can act. So it provides a greater surface area for lipases to hydrolyze triglycerides in those micelles. So just a diagram. So as part of digestion, we'll have a large lipid droplet. Bile salts, which you'll remember are detergents, they have a polar and non-polar part to them. That allows them to bind to lipids and form these small micelles. And you should remember the diagram of what a micelle looks like. There are those bile salts and phospholipids with their polar heads facing out towards the water-based solution. So they're hydrophilic, and then the lipid part of those molecules, or the non-polar part, faces inwards towards the hydrophobic, lipophilic part of the molecules, where there'll be triglycerides, cholesterol, fatty acids, and monoglycerides. Okay, so let's move on to part 2. So the first organ you should have mentioned, going here, is the liver. That's where bile salts are made or synthesized. From there, they move to the gallbladder. That's where they're stored and secreted. The next organ you should have mentioned is the pancreas. And the pancreas produces lipases. So both bile salts and lipases are secreted into the duodenum, that first part of the small intestine. And it's there where they have their action. So the bile salts, as we discussed, cause emulsification of lipid droplets to form micelles. And they provide a large surface area for the lipases to hydrolyze triglycerides to form monoglycerides fatty acids, which remain in the micelle, and then glycerol is obviously produced as well, but that's hydrophilic. It's water soluble, so that goes into solution. So then contractions of the small intestine move these micelles along with the rest of the digestive product, and eventually they reach the ileum. So obviously the duodenum and the ileum, part of the small intestine, and it's in the ileum where absorption of those lipids occur. So this is the next part you should have described. Let's move on to that. So we've got our image of the epithelial cells with the microvilli, and they provide a large surface area over which absorption can occur. So we've got our micelles, we've discussed those. So in step 1, those micelles, if they become into contact with the epithelial cell, the membrane of the epithelial cell, they will release the monoglycerides and fatty acids that they contain, the production of, or a product of those lipases, the action of those lipases on them. So they release those, and they can move into the cell by simple diffusion because they are lipophilic. So they can just pass through the membrane, diffuse in by simple diffusion. That's step 1. So then from there, they move into the smooth endoplasmic reticulum. That's step 2. And there, a condensation reaction occurs to form triglycerides from these monoglycerides and fatty acids. So that's where triglycerides are formed, step 2. And then from there, they're transported to the Golgi apparatus, step 3. And that's where those triglycerides combine with cholesterol, phospholipids, and a specific type of protein called an apolipoprotein. What they form are these spherical particles called chylomicrons. And the job of those chylomicrons is to transport lipids from the diet to the tissues of the body. So supplying lipids from the diet to tissues of the body. That's step 3. And so those chylomicrons then are packaged into Golgi vesicles, and then they're released. So those Golgi vesicles make their way to the cell membrane of the cell that faces the blood, and they're released by a process called exocytosis. So that's step 4. And then from there, those chylomicrons make their way into a lacteal of the lymphatic system. So they move into the lymphatic system, the lymph, and from there, they move through the lymphatic vessels and they drain back into the blood at the thoracic duct in the neck. So that's where they reach the blood. They travel through the blood. They eventually reach capillaries. And in those capillaries, they interact with lipases on endothelial cells that hydrolyze the triglycerides to produce fatty acids and glycerol. And those can diffuse into the cells of the tissue. And so that's a way of supplying those lipids to tissues of the body. So that's step 5. So as I always recommend, if you weren't able to complete that accurately, if you couldn't remember it perfectly, then I suggest you go back and you keep testing yourself on that until you're getting it right, and then you can give yourself more time between testing yourself. Okay, so that's it. Digestion and absorption of lipids.